Next, we would like to invite the Superintendent of International Taiwan University Children's Hospital, Professor Huang Li-Ming, to introduce our second speaker, Professor Michael Wilson. Let's have an applause for Professor Huang.
Good morning, ladies and gentlemen. Welcome to the second talk of the summit. First of all, I'd like to thank the organizers for inviting me to be the moderator of this important talk. I enjoy a lot from the previous talk. That's a very cutting edge. The second topic will be the decoding encephalitis, metagenomic, transcriptomic, and the serologic correlates. This topic will be given by Dr. Michael R. Wilson. Dr. Wilson is a collaborator of the previous speaker, as you have heard from the several minutes ago. Dr. Michael Wilson is a neurologist who specializes in infectious and autoimmune syndrome of the central nervous system. So he's the conditions such as inflammatory condition, demyelinating condition, and also the infection with microbe are the specialty that he has been involved for the previous years. His research involved using genomic technology to enhance understanding of the development of the multiple sclerosis and to identify novel causes of autoimmune and infectious meningoencephalitis. Dr. Wilson obtained his medical degree from UCSF. He completed a fellow neurology resident training at mNGS and Brigham and Women's Hospital. He then pursued a clinical research fellowship in neuroinfectious disease at mNGS and Boston University National Emerging Infectious Disease Laboratories. He completed his postdoc training in metagenomic in UCSF Department of Biochemistry and Biophysics. He belongs to many important organizations, such as American Neurological Association, American Academy of Neurology, International Society for Neurovirology, and the International Encephalitis Consortium. So now please welcome Dr. Wilson to give his talk.
Thank you very much for that kind introduction, and again, thank you to the organizers for inviting us here today. It's a real pleasure. Okay. So these are my disclosures, and I'm going to focus, like Dr. Chu said, I focus pretty exclusively on patients with central nervous system inflammatory disorders, so with encephalitis and meningitis. And in the United States, there are about 20,000 cases a year of encephalitis, and probably a similar number of cases of meningitis. These patients are inordinately expensive to the healthcare system. There's about $2 billion in inpatient costs, and mortality overall is about 10%. And that can range very widely depending on the underlying cause of the encephalitis. One of the hallmarks of these conditions is that, as we've heard, and anybody who's treated these patients recognizes, is that they're incredibly hard to diagnose. It's easy to say that someone has brain inflammation, and it's much harder to figure out why. And so, depending on the study, whether it's in the developing country or in the developed countries, it's typically around 50% of cases go without a diagnosis. There are multiple reasons for that. One of which is we've been reminded of daily for the past year and a half, is that there are many emerging infections, especially emerging viral infections, and many of those have either frequent or at least infrequent neuroinvasive potential. So even if you had a perfect set of pathogen specific tests for the known infections in a particular geography, new infections keep arising, and pathogen-specific or candidate-based diagnostic tests can't anticipate newly arrived infections. A great or sad example of that was the movement of Zika virus into Latin America a few years ago, and phylogenetic analyses of the viral sequences obtained once it was recognized to be circulating in Brazil suggest that it had actually been circulating for 18 months before it was detected, and nearly 2 years before it was recognized as a cause of microcephaly, Guillain-Barré syndrome, and other neurologic diseases that were spiking in Brazil at the time. And that really highlights that, again, candidate-based tests certainly can anticipate an infection that they're not designed to detect. And even once you have those candidate-based tests, if you don't associate a known infection with a particular clinical syndrome, you just won't order the test to investigate that particular patient. So there are two hurdles there. And another reason why encephalitis is difficult to diagnose is an increasing recognition over the last 10 to 20 years that many of the previously thought to be infectious cases of encephalitis we're now recognizing are actually autoimmune. This was a paper from 3 years ago from the Mayo Clinic, suggesting that at least if you live near Mayo Clinic, where they do lots of autoantibody testing for encephalitis, that the prevalence of autoimmune causes of encephalitis is at least as high, if not a bit higher, than infectious causes. And so, as Dr. Chu really well outlined, to tackle the infectious side of the equation, we've worked together for a number of years now to deploy sequencing of spinal fluid and occasionally brain tissue to identify unusual causes of infectious meningitis and encephalitis. And early on, we published a number of case reports and case series, as proof of principle that this technique could be fruitful. The first was a case of a young boy with an inherited immunodeficiency who had a chronic meningoencephalitis that despite his immune deficiency, because such an exhaustive infectious workup had been negative, even in that case, there was a thought that maybe he had an autoimmune condition. But fortunately, in him, sequencing rapidly identified a spirochete infection, leptospirosis, that was very treatable. Another early case report was a patient, an elderly woman who presented first with vision loss, and then strokes, and then a rip-roaring meningoencephalitis, who was found to have Balamuthia mandrillaris and amoebic meningoencephalitis. Another case here of an immunodeficient patient who had a chronic meningoencephalitis over 3 years, and you can see over that time he had very dramatic kind of pan brain atrophy, and sequencing in him identified an orthobunyavirus, Cache Valley virus, that had never been seen on his continent before in Australia. It turned out that he had visited the US 3 years prior before he got sick and brought it back to Australia. Again, an example of where even if people were aware of this viral infection, it wouldn't have made sense to send off the test based on his geographic location. And then another example of, I think, another area for CNS diseases where sequencing could be helpful is patients with chronic meningitis. This was a case of actually a physician, 40 years old, who had a 16 to 17 year relapsing meningitis and arachnoiditis. And she'd been treated multiple times for TB. She'd also been treated for viral infection and for 2.5 years before we heard about her had been on chronic immune suppression for a thought that maybe she had a neurosarcoid-like condition. Sequencing in her identified an infection with Taenia solium. It was neurocysticercosis presenting in an unusual way. And so I think these cases and others highlight the real potential roles for sequencing. One is to identify completely new, highly divergent pathogens, as we saw was done in China in late 2019 with SARS coronavirus 2. Pathogens not linked with a clinical phenotype. So again, a known pathogen, but just that's not thought to cause neurologic disease, or a pathogen that's just not on the differential because it's so rare, like an amoebic cause of encephalitis. And then, as Dr. Chu suggested, with a test, if the sensitivity is adequate, with a test that's able to detect any potential infection except for prions, could it be used as a rule-out test? So if there's already a decent suspicion that a patient might have an autoimmune cause of encephalitis, could a rule-out mNGS assay provide extra reassurance to treating physicians that empiric immune suppression is a good idea and at least wouldn't be harmful to the patient because it's less likely that there's an occult infection that's been missed. And so as Dr. Chu described, kind of a lot of these case reports and case series culminated in this multi-center PIDAID study. And I won't go through all the details of this again, but suffice it to say, it was multi-center, and the idea was to compare the diagnostic yield of CSF mNGS against all the other diagnostic tests sent by the treating physicians. This, again, it was multi-center, primarily in California, but some sites elsewhere in the US, and over 200 patients were enrolled, and it was quite a sick population with half of the patients in the ICU. And again, these are the top line results, like Dr. Chu discussed, that mNGS alone diagnosed 13 cases, and then co-identified cases another 19, and that the cases that were diagnosed only by standard testing fell into those three buckets that I'll talk about in a second. These again are the top line results that were discussed. And these are a couple of vignettes of in the mNGS only diagnoses. One was a case of a dramatic case of a woman with a lung transplant who had had a idiopathic hepatitis for 6 years after her transplant, and then presented with meningitis to the hospital. And sequencing in her identified hepatitis E virus. And not only was her meningitis successfully treated with ribavirin, but her impending liver failure from the chronic hepatitis, which had previously been attributed to fatty liver, that resolved as well, saving her a liver transplant. And then similarly, in another case from the study, an elderly man who was immune suppressed for a transplant presented with encephalitis and was found to have St. Louis encephalitis virus, an infection that hadn't been seen in a human in 30 years in California. In this case, the patient ultimately died of the infection, but I think another piece of the clinical utility of the assay was that the family was able to transition the patient to hospice once they finally understood what the underlying diagnosis was. And I think that's kind of we get very excited about the cases where there's a treatable infection identified, but even in the cases where it's not, we find something that can't readily be treated, there's a great utility to relief to the family and the patient to understand what's made them so sick and to stop the invasive diagnostic testing like more brain biopsies. And so I want to focus on, Dr. Chu mentioned kind of areas where the test, where conventional testing could provide additional information. And I want to talk a little bit more about the low pathogen titer samples. So he mentioned that there were a couple of cases in the study of mycobacterial detection below the reporting threshold. And so we've been had another collaboration with David Bulwer at University of Minnesota and David Maya and his group in Uganda to study patients in Uganda with TB meningitis or suspected TB meningitis. And as we know, this is a highly morbid and very common cause of meningitis around the world. And part of what makes it so morbid and mortal is that it's very hard to diagnose. So culture, CSF culture for TB is ranges anywhere from 20 to 40 or 50%. And then even the Gene Expert Ultra PCR test is about 70% sensitive relative to the a research-based definition for probable or definite TB. And so we have an ongoing study where we're trying to see whether sequencing can enhance detection of TB meningitis, as well as the many infectious mimics of TB meningitis. And so to do that, we're leveraging the specificity of pathogen detection by mNGS, again, both TB and infectious mimics. And then, as Dr. Chu discussed, the idea was that we knew sensitivity was going to be an issue for TB because every test has sensitivity issues for TB because it's paucibacillary. And so we wanted to see whether the host transcriptomics in these CS generated by the mNGS assay could enhance the sensitivity for detecting cases of TB meningitis. And to do that, we looked at spinal fluid samples from nearly 350 HIV infected patients seen in Uganda. And just to focus on the transcriptomic piece, we were very excited that we were able to get a mean of about 3,000 protein coding genes with non-zero counts, transcript counts per sample, which is, is not trivial getting that from spinal fluid, which has so little genetic material. We saw good transcriptomic data even from CSF samples that had low cell counts, which again was exciting to us. And then Akshaya Ramesh, a very talented postdoc, developed a machine learning classifier, first using a training set and then a validation set from these 350 patients to develop a classifier that had a single goal of either calling people as TBM to TB meningitis or not. So in this study, we didn't look at kind of TB versus viral versus fungal versus autoimmune. The goal was just to say TB versus not. And this classifier that Akshay identified would really boil down to 4 genes that had the most explanatory power. And we were excited to see that all 4 of these genes, I won't go into all the details here, but all 4 of these genes have strong support in the literature as being relevant to the T host response to tuberculosis infection. This is the kind of top line results showing that just the machine learning classifier by itself in the validation set had a sensitivity of 83%, but a lower specificity of 76%. And then when we factor in the mNGS data, so the actual pathogen identification to enhance specificity, we get a combined result of sensitivity 83%, specificity 87% for this combined assay. Again, where the data come, both the transcriptomic and the non-human data all come out of the same assay. And then I want to highlight another way that mNGS data can be reanalyzed. This is a paper that just came out 3 or 4 days ago, and this was again led by Wei Gu, who's now an assistant professor at Stanford and worked with Charles and myself, and he reanalyzed CSF mNGS data, again the human part of the data, to ask a question of whether aneuploidy, a hallmark of cancer, could be detected in spinal fluid samples from patients who ultimately were diagnosed with a malignancy. And again, the paper's online, and the upshot was that in 2 different cohorts, including a cohort of patients presenting with suspected infectious meningitis or encephalitis who turned out to have a malignancy, the sensitivity of the assay was 75%, and thankfully the specificity of the assay was 100%, so there were no false positives. So this is yet another potential application of the data of reanalyzing the data from this one single assay. And then I want to talk a little bit about our work that has kind of departed from sequencing because, as Dr. Chu mentioned, sequencing is a wonderful technique, but it's completely dependent upon the presence of the pathogen in the sample that's being sequenced. And as we know, there are many reasons why a virus may only be present for a short period of time. The pathogen may be at a very low titer, potentially because the patient's been partially treated before the sample's been obtained. And so there are reasons why mNGS can provide a false negative result. And so we know in neurology and with patients who have encephalitis, oftentimes the kind of diagnostic test of choice is a spinal fluid antibody test for a particular virus often, or as Dr. Chu mentioned, even for neurosyphilis. And so we've wanted to also develop a companion, more agnostic ability to broadly detect viral antibodies in spinal fluid to see in those cases that are sequencing negative, but for whom a viral infection is suspected, could we find indirect evidence that a virus had been present. And so to do that, we built on a technique published by Steve Elledge and Ben Larman about 10 years ago at Harvard called programmable phage display, also called FIPSeq or phage immunoprecipitation sequencing, and when applied to viruses specifically called VIRScan. And so the idea with this technique is to generate short peptides that span genomes of interest or proteomes of interest, and in this case, many viral proteomes, and to have those peptides expressed on the surface of bacteriophage to incubate those phage with a patient sample that has antibodies, so in our case, spinal fluid, allow the antibodies to pull out phage that are displaying their cognate antigen, and then sequencing those phage to identify which peptides were enriched by the patient sample. And so we built a large VIRScan library. Again, this work was done by Ryan Schubert and Akshay Ramesh, and with over nearly 500,000 peptides spanning all vertebrate, tick, and mosquito viruses. And the kind of proof of principle application was to investigate a condition called acute flaccid myelitis that has been spiking in many countries around the world, including the US over the past few years. And the suspicion has been that this is caused by an enterovirus because of the similarity of the clinical syndrome to polio. But there have been some lingering doubts as to what the potential cause was, because as Dr. Chu's research, there are very, very, very few cases in which enteroviral RNA is detectable in the spinal fluid. So I'll skip ahead here. And so, we put together a collaborative study, including the CDC, California Department of Public Health, and we're able to acquire spinal fluid samples from children with AFM over the three from 2018, 14, and 16, and compared that to the viral antibody profiles of children with other neurologic diseases, and using the VIRScan assay. And the upshot, this paper was published a couple of years ago, the upshot was that the only viral family that had an enrichment of peptides in the AFM children compared to the controls was Picornaviridae, and then that signal was essentially entirely accounted for by antibodies to enterovirus. So this provided more circumstantial evidence that enteroviruses indeed really are the likely etiology in these cases. And this was seen in children who were PCR positive for enterovirus and those who were not. And lastly, as we've mentioned a couple of times, there are patients who have autoimmune causes of encephalitis that may masquerade as infectious conditions. And so to tackle that, we similarly have used phage display to look for autoimmune targets in spinal fluid. And I'll tell you about a case here. So this was a patient I saw in clinic a few years ago. He's 37. He's a man with a history of testicular cancer, but it was in remission for a number of years. And he presented with 9 months of vertigo that had acutely worsened with double vision, and to the point that he had stopped driving. He had to stop working. He wasn't even able to pick up his small children because he would fall over. This is a video of his eye movement abnormalities. You can see that when he, in any direction of gaze, he has a rotatory nystagmus that was, again, quite debilitating. He also has a 6 nerve palsy on one side that was giving him the double vision there on the right. And his spinal fluid was inflammatory at an elevated number of white cells. He had evidence of intrathecal antibody production in the spinal fluid, and there had been a suspicion for a viral infection in him, but everything had come up negative. Because of his cancer history, there was a concern that this could be paraneoplastic, so a tumor-triggered autoimmune condition. And this was his MRI showing some damage to the middle cerebellar peduncle and into the cerebellum. So he was empirically given pulse steroids, to which he had a very dramatic improvement, but then was lost to follow-up for a year because of health insurance issues. And when I was able to reconnect with him, he'd had further worsening of his neurologic condition and radiologic worsening to the point that he had a suspected brain tumor, and they were preparing to do radiation. But what we informed him was that in the meantime, we'd been studying his spinal fluid and identified that he had antibodies to a protein called Kelch-like protein 11. This had been validated in a number of ways and has been published. And what was fascinating was that it turned out that colleagues at Mayo had been collecting patients like this, men with ataxia and a history of testicular cancer for 20 years, but hadn't identified the autoantigen in their case. We informed them of the antigen in his case, and it turned out that all these men at Mayo also had a similar had antibodies to the same target. So this is a newly described paraneoplastic neurologic syndrome that causes a brainstem and cerebellar inflammation. So I'll stop there just and conclude that, as Dr. Chu highlighted, that sequencing of spinal fluid is now clinically available, has a rapid turnaround that does increase diagnostic yield in patients with meningitis and encephalitis. It can have a real benefit both in terms of treatment, but also in terms of closing the diagnostic odyssey that many of these patients go on. In addition to just identifying a single pathogen, there's a rich amount of information, additional information that comes out of sequencing in terms of genotyping a virus, identifying a particular bacterial species, resistance mutation identification, and the subthreshold reads that Dr. Chu discussed. We know that mNGS, like any diagnostic test, has blind spots. So for compartmentalized infections that are better serologically diagnosed, infections that are very low abundance in the spinal fluid, and then in cases where the pleocytosis in the CSF is very high, the human sequences can decrease the sensitivity for pathogen detection. We didn't get into it today, but I think even in sterile fluids like spinal fluid, there's a real critical need to have rigorous controls to rule out and to filter out environmental contaminants. Spinal fluid sequencing looks as every spinal fluid that's sequenced looks as it has bacterial meningitis, but we know that these are contaminants from the laboratory and from skin flora on patients that are picked up when people have a lumbar puncture. As we've both highlighted, there are really exciting opportunities for host-based expression, signature analyses, both in neuroinflammatory syndromes as well as systemic disease. And then again, this recent study that's just come out showing that with the same data set, there's potential to identify tumors that may be masquerading as inflammatory conditions. And then I've shown you a little bit about our work with antibody profiling as a backup to direct detection tests like sequencing, both to identify viral infections in the CNS as well as autoimmune conditions. There are many ways to identify antibody targets. Phage is one that's that we like because it's fast and comprehensive, but it has its own limitations where it's not good at picking up conformational epitopes or epitopes with post-translational modifications. And I also want to thank a huge number of people in my lab and Joe DeRisti's lab and Charles Chu's lab. This is, as you can tell, a lot of this work is highly collaborative and very fortunate to have such great colleagues, and also to our funders. And lastly, to our patients. This is the young boy with leptospirosis who's now in college. Maybe he's done and has done quite well. This is a patient who had the Cache Valley virus, again, who did not survive the infection, but again, his family was happy to know that at least what had made him so sick over a number of years. So thank you.
Thank you, Professor Michael Wilson, for your kindly sharing your studies. And the same, leave the questions under the panel discussion session. So thank you, Professor Michael Wilson. And currently we have around 500 attendees online, and we'll have a short break for about 10 minutes. So probably we can come back at about 10:15. So thank you for the participation in the first session. Thank you.